Archiving started.
In this presentation, we will look at a few of the digestive system slides. The first one will be of the parotid gland, and you remember that the parotid gland is the largest of the three pairs of salivary glands. So on this first image that you see, this is a scanning view of a parotid gland. Now, I want you to go in your lab book in exercise 43 and look at the written description of the salivary gland. Now, it will call it salivary. It may not call it parotid, but this is the parotid gland. And notice in your book that you're going to look for some distinct ducts as well as some little secretory clusters. These secretory clusters are the cells that actually will produce the ingredients of saliva. Some of these are mucus secreting clusters, some are serous secreting clusters. I just want you to be able to see the clusters themselves. So all of these areas, all of this, you're seeing various clusters of cells. But then if you look at what I will encircle here, you see a distinct duct. And I can tell that's a duct. When you put it on a higher power, you will actually see the cuboidal cells. Remember from AMP1 that cuboidal cells line ducts? Here's another one up at the top of your screen. Right, so at first glance, this will look quite a bit like a pancreas slide, because in the pancreas, that's another exocrine organ as far as pancreatic juice is concerned. But when we saw it before, we saw it as an endocrine organ. It looked a good bit like this with the addition of some islets of Langerhans, which you won't see on this slide. Okay, if we move to the next image, let's wait till that comes up. All right, here you're seeing it a little bit closer. Right here, now you can see it. It's on a higher view, and here you can see a duct. Here you see another duct. All right, and then mostly what you're going to see are these secretory clusters. So there and there and there, pretty much everywhere else you see secretory clusters. I also see some adipose cells. Do you see those in there? And you see some fibrous tissue that separates the lobules that are here. But for your test, if we move to another slide, let's look at this again. But again, for your test, you can tell that this is a salivary gland just for this section, because you see the secretory clusters as well as some distinct ducts. Do you see any ducts in this slide? Do you see one there? There's a duct. Now here, that's, well, let me move that back. Hang on. Ignore that last one I did. Let's start over. Okay, there's a duct. There's a duct. And then here you see another duct, but you can see pieces of it just above and to the left of it. That's almost a lengthwise section of that duct. Here's one on the lower left side of your screen. Here's a large duct, and here's several right here. So these are the ducts. Make sure you can identify those. And then you also see the secretory clusters. Now, if we move on to the next view, it takes it a minute for these to load. I apologize for that. Eventually, it will get there. Here we go. Now you can see it on high power. And the ducts are much more obvious here. So there you see a duct. Here you see another duct. And then you see several secretory clusters. All right, in this area, with this secretory cluster, notice that the cytoplasm of the cells is fairly light in color. Those are some mucus secreting. In this one, you see a little bit darker, it's grainier looking, and that's probably some serous secreting cells. If we move to another slide, be patient. Okay, here you see just another view of it on the higher view. Notice you can see the duct. That's real easy to see on your right-hand side, excuse me. But look at the various other clusters. You see that the cytoplasm stains differently in some of the cells. That just has to do with what chemical component that cell produces for the saliva. You know, you have enzymes produced in saliva, you have bicarbonate ions, you have mucus ions. For the lab, you don't have to know the difference in those cells. Just know those as secretory clusters. And then again, you have the ducts there. So this one's pretty easy to recognize if you look for the two distinct regions, the clusters and the ducts. All right, on the next few slides, we will be looking at the esophagus. And if you'll notice, on the esophagus, lining it is going to be the non-keratinized stratified squamous epithelium. So you should see it in view now. At the top, I'll just draw a line to go through the width of it. That's the mucosa, what you're seeing with this blue line. There's the mucosa. And this looks quite a bit like a slide you may have seen from A&P1 when you were looking at the stratified squamous epithelium, because that's what the mucosa is made up of. It's very important that it's that tissue because it's a thick tissue, and that helps prevent things from absorbing through the wall of the esophagus. You won't see this in the lower parts when you get down to the stomach and the intestines, because those are absorbing organs, so you wouldn't want a lot of layers of cells covering that inner surface. Okay, so that would be the mucosa that you see in blue. And now I'm going to, we'll look into the submucosa, and if you look at this area right through there and right through here, those are some mucus glands that are down in the submucosa. And over here, let's see if I can, in that area, that's probably a large duct that those mucus glands are emptying into. And you don't see where it goes up onto the surface, but it's got to be sectioned just right for that. But for the lab recognition, you need to recognize that in the esophagus, you're going to have the non-keratinized stratified squamous epithelium for the mucosa, and then within the submucosa, you might see these lightly stained mucus glands. So if we look at some other views on this same slide, this one's on scanning. The next slide, I believe, is also going to be on scanning. It's just a different area of that same slide. You can see, again, the non-keratinized stratified squamous epithelium on a higher power, which we'll see in a few moments. That will be more easy to recognize. And then in the submucosa, there's some of that right there. You see the mucus gland. And then you see mucus gland doesn't cover every bit of the submucosa. Submucosa would run through there. Excuse me. Down below the submucosa, I'll put an X in this area. That's part of the muscularis mucosa. All right, let's move to another view. All right, now here again, you see your mucosa. You have your, oh, let me erase that. Okay, now you're seeing a higher power. And here you really get the idea that it's the non-keratinized stratified squamous epithelium. You can tell it's non-keratinized because the cells at the top still have a nucleus. I see little nuclei here and there. All right, so that means they're living. They're not keratinized. But all of this area right there, that makes up the mucosa. And then the rest of what you're seeing is submucosa. And within the submucosa, do you see right through there? I've circled part of one of those mucus glands. Okay, if we move to another slide, still showing the esophagus, it's just a different view. Just give it a moment to move. I don't know why this is so slow. Here we go. Now this is just another view on the same slide. It's not positioned just exactly the same way, but you can see sections of the mucus gland right there, a little bit right there, and then this area, that would be a duct that is probably draining through. Over on the left-hand side of the screen, this is the mucosa. So you're seeing the stratified squamous epithelium. Okay, I think that will do it for esophagus slides. The next few slides will be of the stomach. In your lab book, if you will read about the mucosa of the stomach, it will tell you that there are tiny little glands in the wall of the mucosa that produce gastric juice. Those glands are called gastric glands, and that's how you're going to recognize that you're looking at a stomach slide. Here on this slide, the mucosa, remember the mucosa is the lining. There's the mucosa. and then the submucosa is underneath that. There's the submucosa. And then in the stomach wall, the muscularis mucosa is very thick. There's 3 layers of muscle. But what I want you to focus on for your lab is just the mucosa. And within the mucosa, you see these little openings into these glands. And those openings that you're seeing there are the gastric pits. I want you to notice on this that the surface of the mucosa is somewhat uniform. We're going to see in the small intestine, it's not going to be nearly so uniform there. But make sure you can identify the little openings into the glands. Those are the gastric pits. I will erase this and try to draw a line within the gland. So that would be a gastric gland. There would be one. You don't see them on a perfect section here, but it's all through the mucosa. You've got a drawing in your lab manual that shows this very well. Now, if we move to another slide. Okay, here you see the same organ, but it's just showing it from a different view. I've moved to another part of the wall, and so you're seeing there's your mucosa right there. And so, again, you're going to see the gastric pits. Right, so there would be a gastric pit. There would be a gastric pit. And those lead into the gastric glands. Right? If we look at another view, I believe this next one's on a higher power. These first 2 slides were on scanning. And now this is low power. Remember, you start with scanning and then you move to low power. And on this, the gastric pits are real obvious. So you can see those gastric pits. But still, the surface of this is fairly uniform across there. You can see that. The gastric glands, you can see all through the mucosa in here. You can see all of this is gastric glands. Look at the drawings in your lab book and in your textbook, and you'll see those gastric glands more easily. If I move to another slide of the stomach, if we wait till it moves, I'm sorry again, it takes so long to get from one to the other. Even though this is positioned differently, you still should be able to see the gastric pits on the left that lead into the glands. This is sectioned real well, so it's pretty easy to see the glands. On the far right of your screen, over here, this is part of the muscularis mucosa, that's smooth muscle fibers. And then in the middle, in these areas, this would be in the submucosa. The mucosa of the stomach is pretty thick. There it is right there. All of that is the mucosa. And it's made up of these very, very, very numerous gastric glands. Now, this last slide of the stomach is going to show the gastric glands. You can see just some of the details of the cells of the gastric gland. If it will move, I've clicked it. Here we go. Now, I know I don't expect you to know it from this magnification. You need to be able to recognize it by seeing gastric pits. But just for extra information, and this especially goes along with your lecture, you might recall that within the gastric glands you have chief cells and parietal cells. You can actually see the difference in the two on high power with the microscope. The chief cells are ones that you see right here and right here. Notice that their cytoplasm is more of a bluish color. Now, they all have a bluish colored nucleus, but look in the cytoplasm. See how it's a little darker here? Those are chief cells, and they produce enzymes like pepsin. On the other hand, the parietal cells have more of a pink staining cytoplasm, and they tend to be a little bit larger. Those cells produce hydrochloric acid. So here we see some on the right. And then on the very far right bottom corner, you see quite a few chief cells. So the parietal cells are pink, and that's easy to remember. Pink and parietal start with a P. And then the chief cells are the bluish stain. Again, you don't have to know the difference in chief and parietal cells for lab. For lecture, you learn their difference, not with what they looked like, but what chemicals they produced. All right, that's it for the stomach. The next few slides will be of the small intestine. The particular section of the small intestine that these slides are showing is the duodenum, which is the first part of the small intestine. So as it moves, we'll see it on, this is on scanning. Now, earlier, just a few minutes ago when we were talking about the stomach, I mentioned to you more than once that the surface of the mucosa in the stomach is rather uniform. The surface of the small intestine is not uniform. See how it's real bumpy looking? It goes down and up, and you see parts of the mucosa jutting out. See how that juts out? How this juts out? Let me erase some of these. Do you see how this juts out? In your lecture, you learned that these finger-like projections, which here you see it in pieces. It's got to be sectioned just right to show that. But those are called the villi, and they extend out into the lumen. The lumen is this area. That's the opening inside the organ where the food is. And how you recognize a small intestine slide is by the presence of these villi. So make sure, you know, this is a villus, this is a villus, this is a villus. Here's just a section of one. There's a villus, there's a villus. The entire mucosa runs from the tip of the villus all the way down to that point. And then in the submucosa, you see some glands that look a lot like those mucus glands in the submucosa of the esophagus, because they are mucus glands, but these have a special name. They're called Brunner's glands. So you'll see that in the other written descriptions that I've got for you in the folders on these slides. But you see the Brunner's glands there. Deep in the mucosa, you also see some little glands that would be analogous to the gastric glands in the stomach. But in the small intestine, they're called either intestinal glands, or the older name for those, it's kind of a funny name, it's called crypts, C-R-Y-P-T, crypts of Lieberkühn. And those are producing intestinal juice, just like gastric glands in the stomach produce gastric juice. So for the small intestine, you see, in the mucosa, you see the villi going out into the lumen, and then you see the intestinal glands dipping down into the mucosa. Okay, the Brunner's glands actually empty into the intestinal glands, so mucus would also be a secretion in this intestinal juice. Here's another view. I believe this one will still be on scanning, but it just shows a different area. Here you easily can see the villi. Those are the finger-like projections into the lumen. I'm seeing right here, you see the crypts of Lieberkühn, and then in the submucosa, you see the Brunner's glands. On the next slide, if it will move on, here it is. You see on now, instead of scanning, you see this on low power. You can easily see the villi. There's a villus, there's a villus, there's a villus, there's a villus. And then down here, deep in the mucosa, you see crypts of Lieberkühn. Okay. I want you to notice on the surface of each villus, notice the cells that are there. When we put this on a higher power, oh, look right here. Look at that. That should be pretty obvious. That's simple columnar epithelium. The little holes that you see, you might remember from A&P1. Remember goblet cells? I'm going to put the arrow onto the openings within goblet cells. You will see this in the small intestine. You also may have noticed this if you looked at the surface of the inner surface of the stomach on high power. You would also see the simple columnar epithelium. Remember, that's an absorbing epithelium. That's a major tissue of the digestive tract, because the digestive tract not only digests, it also absorbs. So here's another view on the high power. Again, here you should be able to recognize, like right here, you can easily see that simple columnar epithelium. You see it there. It's got to be sectioned just right to see it perfectly. The little bubbles that you see within it, that just represents the goblet cells, like there, there. Remember, goblet cells produce mucus. On this next slide, All right, here you see it on high power, and you actually can see the columnar epithelium. See all of this is columnar? And I want you to notice on the surface of it, see how it looks real hazy? It almost looks like somebody took a magic marker on the surface. There you see on the other side of this other villus. This represents microvilli, not cilia, like you would see in the respiratory tract, but microvilli. So you might want to go back and look in your textbook for lecture. Look at the significance of that microvilli. It literally just increases the surface area for absorption. You only see that in the digestive system with the small intestine because that's the organ that absorbs the most. The last slide we're going to look at in this section is of the large intestine. Remember, another name for the large intestine is the colon. The colon produces massive amounts of mucus, and these slides have been stained specifically to show the mucus as a bright pink. So you see all those dark pink dots, those bright pink dots? That's actually mucus that's produced by goblet cells within the mucosa. There are lots of goblet cells in these glands that at first glance this may look somewhat like the stomach because the surface, again, is rather uniform, but the gland itself pretty much goes just straight down, and it is loaded with goblet cells. And so that's how you know this is the large intestine. It doesn't even have to be stained to show the goblet cells. If you just saw a straight tube that had a ton of holes in it, those holes would be where the mucus was. Okay, so here you can see all these pink dots that you see. That's the stained mucus of the goblet cells that line the tubular mucus glands that are so prevalent in the mucosa of the large intestine. So this one's very easy to recognize. Okay, that's it for the digestive system slides. Of course, there are some other digestive organs. There is a pancreas, there is a liver, but I don't have those for you on a test or a quiz. So for digestion. So if you'll make sure you know the salivary gland, the esophagus, the stomach, the small, and the large intestine, that would be great. Make sure you look at the other slides that I've got in your slide folder besides these in the recording. Make sure you can recognize everything that's identified on those slides. Also look in your lab manual in exercise 43. There's a very thorough description of each of the slides.
Archive recording.